Hi, this is Dr. Lauren Launen, and this is the last video in a video set that together comprise a lecture on DNA synthesis in cells. We're going to focus in this last video lecture on telomeres, which address an interesting problem that occurs on the end of eukaryotic chromosomes. Only eukaryotic chromosomes because they are linear, not prokaryotic chromosomes, which are circular. So I'm going to remind you about what you should already know about DNA synthesis, and an important aspect of that is that all new synthesis occurs 5' to 3', adding new nucleotides to the 3' end of the growing strand. So in this figure, if in black here is the parental or template DNA here at the top, if this is the 5' end, that means this is the 3' end. That'll mean that on this new strand that is shown up here, the newly forming strand, this is going to be the 5' end, and this will be the 3' end. So the arrow is correctly indicating the direction of synthesis. The blue is the area where there was an RNA primer, and the red is the new strand, and it's the leading strand in this case. All right, that's fine. Now let's look at what's happening down here on the lagging strand. Here's the 3', and here's the 5' of the template for the lagging strand. We're saying that this is the lagging strand, whereas up here was the leading strand. And lagging strand synthesis, as you should already know, involves discontinuous synthesis of Okazaki fragments, short bursts or pulses of DNA synthesis, where all of the protein machinery is working over and over and over again. So here we see the RNA primer, and then a length of DNA, an RNA primer, and the length of DNA, and so on. And you should remember that what happens to RNA primers in new strands of DNA is that the enzyme DNA polymerase I, discovered by Arthur Kornberg, is going to come in and take that RNA out and replace it with DNA, with the enzyme ligase sealing up the phosphodiester linkages at the edges. So here we've got a gap in the lagging strand synthesis, and then it has been down here, it has been completely filled in by the combined action of DNA polymerase I and DNA ligase. But look at what's happening over here on the end. Here we've got a gap that's created when this RNA primer has been removed by DNA polymerase I, but then poor DNA polymerase I is looking for let's just draw it in here. It's looking for a 3' hydroxyl group right there, right in this spot, but it's not finding it. And so, because it's not finding it, DNA polymerase I cannot put DNA in this region, leaving a permanent gap at this end on the lagging strand. So what will happen over time is the leading strand will be made correctly, the lagging strand will always have a gap on the end. And as the cell goes through round after round of DNA synthesis, each time cell division occurs, there will be a gap there. And every time that that happens, it means that this chromosome will in fact get shorter. So the DNA sequence on the ends will just get chipped away a little bit at a time with each round of DNA synthesis. Well, what does that mean? That means that essentially your chromosomes are much longer when you are young than they are when you are old, after you've gone through many rounds of cell division and DNA replication. So that's got some problems associated with it. If the DNA that is lost on the ends over each round of cell division is essential DNA or coding DNA. And so the solution to that is that eukaryotic cells evolved a long time ago to have these chunks of repetitive DNA on the ends of the chromosomes that are called telomeres. And telomeres are there simply to protect the ends because they are DNA that can be safely chipped away over time, leaving the important chromosomal content intact. So think about this a little bit. Here we are age 25 and cell division is occurring, and this is sort of progressing through like a human, to maybe a typical human lifespan if you're lucky enough to make it into your 70s. There are your chromosomes. They're drawn the way they would look as you're going through mitosis or meiosis perhaps, not just in their single linear form, but in that sort of X structure that's only going to happen during mitosis or meiosis. But anyway, here are the long telomeres on the end. They get chipped away a little bit shorter still, and then once you reach the older age, towards the end of your lifespan, there are almost no telomeres left. And that's when you start chipping away into the essential material of the chromosomes, and in fact, this phenomena is associated with aging. So your telomeres are much longer. You're closer to age 25. Me, I'm getting pretty close to 50, so mine are starting to be a little bit chipped away, and the clock is running out for all of us. So how do cells do this? They build themselves telomeres using an enzyme, really interesting enzyme called telomerase. And telomerase is only active in embryonic cells. Now that's what's normally the case. However, telomerase is active in some cancer cells, and that's one of the reasons that cancer cells are quote-unquote immortal. When we take them out in cell culture, they can keep dividing and dividing and dividing over many, many rounds of cell division, basically with no limit. So the famous HeLa cells from Henrietta Lacks's cancer back in the 50s, those cells are still, their descendants are still alive today and used in cell culture, and that's because the enzyme telomerase is active in those cells, as it is in other types of cancer cells, and the telomerase is responsible for rebuilding telomeres. So here, this is there's a kind of tiny font here, a little bit hard to see, but here this green blob is the enzyme telomerase. And the enzyme telomerase, by the way, we could get into a lot more biochemistry on it. It actually has an RNA template built right into it. It's right here. So it's a lot like a ribosome in that telomerase is a combination of protein subunits and a ribosome, or an RNA molecule. So here's the RNA molecule, and it is such that it will actually base pair to the end of the overhang. This overhang here, this overhang right here, telomerase will base pair to that in embryonic cells, and when it base pairs to this end, you then have a 3' hydroxyl group, and the enzyme is able to catalyze addition of nucleotides to the end of that, extending the overhang, making it a little bit bigger, and then binding to it and synthesizing in the opposite direction on the new strand, filling the gap that's been created in. And it will do this over and over and over again. It'll keep binding to the end, and then it'll backfill, and then it'll bind to the end, and then it'll backfill, and so on. So that is a little bit of information about telomeres. So telomeres are segments of repetitive, non-coding DNA on the ends of the linear chromosomes found in eukaryotes. Telomeres are there to protect the ends of DNA from the continuous shortening that always occurs during DNA synthesis, which precedes each round of cell division or cytokinesis. And telomeres are made by the enzyme telomerase that should only be active in embryonic cells, embryonic stem cells. However, it's also active in the immortal cells that are known as cancer cells. And that concludes this lecture.